Cell structure, AQA biology. Key terms. Well, magnification is the size of an image divided by the size of an object, which is the actual calculation. And it's how much bigger an image is to the actual specimen. Then the resolution is the minimum distance apart that two objects can be seen as two separate objects. Because at a certain magnification, two objects that are very close together will start to look like one. So the better resolving power, the better in the microscope. Methods of studying cells. You have microscopes. And how microscopes work are that individual cells are too small to be seen with the naked eye, so you use a microscope instead. The maximum magnification of an optical microscope is 1500. The maximum resolution of an optical microscope is less than 250 nanometers apart. Electron microscope. Now, there are two types of electron microscopes that you need to know. You need to know about TEM and SEM. You don't really need to know what they stand for, to be honest. They really won't ask you. But TEM uses a beam of electrons, and it has a shorter wavelength than SEM's. It can magnify objects by 500,000 times and has excellent resolving powers. SEMs scan over the surface of the specimen and a 3D image is obtained, and it's a slightly lower resolution than TEMs. Advantages and disadvantages of electron microscopes. Well, an advantage is that you have a higher resolution, a higher magnification, and a shorter wavelength, so you get a better image. A disadvantage is that it's easily deflected because it uses a beam of electrons, which are very small particles. It has to be done in a vacuum, which means no living specimen can be viewed. A fluorescent screen is needed to see the results, and due to the harsh procedures, it could alter the structures of the internal organelles. Microscope measurements and calculations. Well, the eyepiece in the calculation, most of the time is magnified by 10 already. So when they're talking about sometimes in a question, they might say an image is magnified by 50 times, but the eyepiece had a magnification of 10. How big was the image magnified by? So it would be 500, because you actually have to times the eyepiece magnification by the magnification used to see the specimen. And this is the eyepiece, so you know exactly what I'm talking about. Now, eyepiece usually has a scale and has an eyepiece Grant tool, and this has 100 divisions, which is typically about 10 millimeters long. Now, you can use this to measure internal organelles, because you're not going to exactly be able to take a ruler to that and measure it. And this is a calculation for magnification. I remember it as I am. So I because I'm at the top, and then am, which is the other two. So I is the image size, A is the actual size, and M is the magnification. Now, thank you for listening to this video. This was all about microscopes and cells and such. And the next video will be about cell organelle structures, for instance, like nucleuses and stuff like that. Thank you for listening.